Hello and welcome, students. Today, we're stepping into our lab where we will be actively exploring the fascinating world of plant hormones with a hands-on practical. So far, we've delved into how plants respond to their environment using hormones. You've learned about phototropism, where plants grow towards light, and geotropism, where they grow in response to gravity. We've specifically looked at the role of auxin, a crucial plant hormone that either promotes or inhibits cell elongation, depending on whether it's in the roots or shoots, guiding plants to grow towards or away from stimuli. Today's practical will build on that foundation by giving you direct experience with the function of auxin. By conducting these experiments, you'll deepen your understanding of how this hormone can be manipulated to benefit plant growth and crop yields, aligning with what we've learned about plant responses to their environment. This will provide you with a practical perspective on the theoretical knowledge you've acquired. Before outlining the practical, let's take a moment to consider its importance. Understanding the functions of plant hormones like auxins, gibberellins, and auxin allows you to appreciate the tremendous impact these hormones have on plant growth and development. Their roles in processes such as seed germination, flowering, and fruit ripening are not just academic. These hormones are actively manipulated in gardening and farming to optimize plant health and crop yields. By grasping how these hormones work, you gain valuable insights into practical applications that can improve agricultural productivity and sustainability, potentially impacting food security on a global scale. Second, investigating plant hormones in a practical setting sharpens your experimental skills and reinforces the scientific method. You'll learn how to design experiments to observe the effects of these hormones on plants, how to accurately collect and analyze data, and how to draw meaningful conclusions from your observations. Mastering these techniques now will also enhance your problem-solving capabilities and analytical thinking, which are invaluable in any discipline. Finally, this practical encourages you to think critically about how different factors influence plant growth. By experimenting with plant hormones, you are prompted to hypothesize, observe, and interpret how these hormones can be used to achieve desired outcomes in horticulture and agriculture. This process fosters a deeper understanding of plant biology and instills a scientific approach to problem solving. You'll be able to apply these analytical skills in various contexts, from optimizing your home garden to participating in large-scale agricultural projects. Let's dive into the fascinating world of auxins, gibberellins, and auxin and discover how they can transform our approach to gardening and farming. Our first step is preparation. We begin by setting up a clean and organized workbench with all the necessary materials. 3 Petri dishes, 10 cress seeds per dish, moist cotton wool, a ruler, and a light source. Ensure that the cotton wool in each Petri dish is evenly moistened to provide a consistent environment for all seeds. Second, we need to set up the experiment. First, place 10 cress seeds into each of the 3 Petri dishes lined with moist cotton wool. Then place 1 Petri dish under direct light from above. Position the second Petri dish in an area with partial light. And then store the third Petri dish in complete darkness. Now, for our second setup. We will again place 10 cress seeds into each of 3 Petri dishes lined with moist cotton wool. Shine light directly above the first Petri dish. In the second Petri dish, direct the light source to the left. For the third Petri dish, direct the light source to the right. Our third stage is observation and recording. Over the course of 1 week, measure the height of each seedling in all Petri dishes daily. Use a ruler to ensure accurate measurements. Record the heights in a table for each day, noting the variations between the different light conditions. You will need one of these tables for each Petri dish. This one is just for Petri dish 1, which is in direct light. Also, make sure that the temperature and moisture levels remain constant throughout the week. This ensures that the only independent variable is the light. The fourth stage is data analysis. calculate the mean height of the seedlings in each Petri dish at the end of each day. This will help compare the growth rates under different light conditions. For the second setup, create a labeled diagram illustrating the direction of growth of shoots and roots in response to the light source. Now, let's consider the visual differences in growth. As the week progresses, the differences in growth depending on light conditions will become visibly apparent. The seedlings in the dark may exhibit pale, elongated growth, whereas those in direct light may appear robust and healthy. The second setup will show a clear phototropic response as the seedlings bend towards the light sources from the left or right. This will offer a satisfying visual demonstration of how plants grow directionally towards light. Watching the seedlings respond differently to varying light conditions offers a fascinating insight into the role of plant hormones in growth and development. Let's now look at this experiment in more detail. The central biological reaction in our practical involves the response of plant hormones, particularly auxin, to different environmental conditions. This process, known as phototropism, helps plants optimize their exposure to light for photosynthesis. Let's recap the key steps of this practical and their importance. First, why did we place 10 cress seeds in different Petri dishes? Well, we did this because ensuring each Petri dish has 10 seeds standardizes the initial conditions. It helps in achieving accurate comparisons by providing consistent data points for analysis. Second, let's consider how we control the lighting conditions. By varying the light conditions, directly above one dish, partial light for the second dish, and darkness for the third, we can observe how light direction and availability impact plant growth. This step is crucial for studying phototropism and understanding how plants grow towards light. Third, let's consider how we can maintain a consistent environment. Each Petri dish is lined with moist cotton wool to provide a consistent and optimal environment for seed germination. Controlling other variables like temperature and water ensures that the observed differences in growth are due to light exposure. Fourth, why did we take daily measurements? Well, measuring the height of each seedling every day allows for tracking growth patterns over time. Recording this data helps identify trends and makes it possible to calculate daily growth rates for each condition. Fifth, calculating the mean height of seedlings in each Petri dish each day helps in reducing individual anomalies and provides a clear indication of the overall growth trend. Mean values are useful for comparing the effects of different light exposures. Now that we have all of our results, we can analyze them. In our analysis, we should discuss the impact of light on growth. For instance, expect the dish in full light to have the tallest seedlings as light promotes growth. The dish in partial light might show some bending towards the light source, while the dish in darkness could exhibit elongated but weak seedlings due to lack of proper light. Throughout the experiment, we must ensure that all other conditions like seed type, temperature, and water supply are consistent across all Petri dishes. Any variations here could lead to inaccurate conclusions. Finally, we should evaluate the setup for any possible errors or deviations. Consider factors such as uneven light distribution, differences in moisture levels, or any other uncontrolled variables that could have impacted the results. By carefully analysing these steps and considering the impact of control variables, we can draw conclusions about the role of auxins and other plant hormones in growth and their response to different environmental conditions. This practical helps reinforce the understanding of plant hormones and their crucial functions in growth and development. In today's lesson, we conducted a practical experiment to understand the function of auxins in cell elongation. Our key takeaways for future practicals are to precisely control external variables such as light and temperature to ensure reliable and repeatable results. Carefully apply hormones to avoid contamination and ensure accurate observations. And finally, understand the specific applications of each hormone in agricultural and horticultural practices, noting how they can be manipulated for desired plant traits. This practical not only deepened our understanding of plant hormone functions and applications, but also highlighted the importance of controlled experiments in studying plant biology.